Welcome to Inside the Paper, an EndosKool series where we talk directly to the source. In this segment, we don't just summarize the study. What we do is we sit down with the author to unpack what the research really means, what led to it, what the data truly shows, and also how it may change the way we think about clinical practice. Today, I'm joined by Dr. Prasanna Neelakantan to discuss a fascinating paper on the intracellular bacteria in stem cells of the apical papilla, and what this could actually mean for regenerative endodontic failure. The study introduces a provocative idea that bacteria may survive inside the very stem cells that we rely on for regeneration, acting almost like a Trojan horse. So today we're going inside the paper with Dr. Neelakantan. Dr. Neelakantan, thank you so much for joining me.
Thank you so much, Alan. It's a pleasure to be here, and I'm honored you chose our paper for this.
No, it's terrific. I thought it was a unique idea, and it's a tremendous finding. So I figured it'd be a good thing to share with our audience. First and foremost, what inspired you to research this topic and to study it?
All right. So before we go into that, I think I'd like to give a brief background of what we do in our lab here. So my research lab's focus is primarily studying biofilms and antimicrobial therapies, and we do this both with endo and non-endo contexts, not just root canal systems. And we look at skin infection models, oral mucosal infection models, and how bacteria or microorganisms in general can evade our host responses. And as endodontists, we all know regenerative treatments are successful when you look just at the mineralization standpoint or the root lengthening standpoint. But does true regeneration ever happen? And we know the answer is no. There's abundant evidence out there which tells us it does not happen. And there's also quite a lot of published evidence talking about how infection can alter stem cell behavior. We know that bacteria can alter stem cell behavior. And people have published, the San Antonio group has published a lot about this. They've spoken about how bacteria can influence stem cell behavior, how bacterial toxins can go and influence behavior. But what people have not spoken about much, which is well characterized in many other sites in the body, like skin infections, for example, or in wound infections, is how bacteria could go and hide inside our own cells and use it as a reservoir to potentially multiply and cause an infection. Now, is this very new in endodontics? No. If we look at the literature, many decades ago, Walton and Arjman reported this, and they showed in monkey models that in periapical biopsies, they could see bacteria inside macrophage cells. Yeah. And that was something which triggered, and I said, why have we not considered that as a failure mechanism for even persistent apical periodontitis? We speak about infection, toxins, all that, but we've never really explored that finding that intracellular bacteria can sustain periapical infections. And then when we had the discussion, I had a discussion with my PhD students who are not dentists. They are all from the biotechnology background. And then we discussed, what about other cell types? Can bacteria ever go into stem cells? Could we just test that out? So I had my student her name is Shantani, one of the best trainees I've ever had. She's a biotechnology graduate. So she went to the lab and I told her, just take these stem cells, try if bacteria go into them and come and tell me what happens. So she went to the lab, she tested and she comes and shows me the data. And then I look at that and say, something doesn't look all right. Because if the bacteria are going into the stem cells, we know they should do one of two things. One is the bacteria should kill the stem cell, or the stem cell should design some approach to kill the bacterium. But what we were seeing is data over about 14 days where the bacteria are not dead, the stem cells are also not dead. So I told her, let's repeat this because I want to be sure, because you're opening something which is going to be a Pandora's box. Mm-hmm. So go back and test it at least 2 or 3 more times. So she goes and repeats it, and we get this consistently that when we take stem cells of the apical papilla and challenge it with bacteria, The bacteria don't go kill the stem cells. The stem cells are not developing immune mechanisms to kill the bacteria, but both are alive over a 14-day period. So I said, okay, now let's start profiling what's exactly happening. How are they going inside? And once they go inside, what are they doing? So the bacteria go inside, they don't multiply, they just remain sort of metabolically inactive for quite some time. And they likely shut down some pathways in the stem cells, right? So we had this finding. So I said, okay, so what are implications clinically? So we know it goes in fine. They're not dying, okay, but how is the function of the stem cell being affected? Because we know that we want our stem cells to biomineralize or differentiate in different cell types. So we go back to the lab and then we look at these mineralization assays, and we find that when bacteria are inside these stem cells, they do not allow for the stem cells to do any kind of mineralization. So you have complete abrogation of dentine formation. And that was exciting. So then we said, okay, we went to the Foundation for Endodontics and submitted for a grant application and said, hey, we want to study this in greater detail. And the foundation is amazing in funding studies that are so innovative, and we got a full funding on that one. And then we went in deeper to study about what kind of genes are being regulated, what exactly happens. And we found that what happens is that when bacteria go into these stem cells, they shut down all those mechanistic pathways that stem cells use to produce dentine-like mineralization.
So differentiation is kind of affected.
Differentiation is abrogated. And I think this has implications beyond just regenerative endo, because we know that when you have a biofilm inside the root canal, organisms can definitely exit the canal space and go into the periapical tissue region. Although your host immune response is likely to kill most of these organisms, there'll be some bacteria which will evade the host immune response and reside into the periapical cells. People have shown this in periodontology. They've shown that bacteria can survive inside periodontal ligament fibroblasts, just that no one's shown what happens to stem cells. So this could very well disclose or uncover a mechanism for treatment failures as well as persistent apical periodontitis beyond just regenerative approaches. And in terms of what we do from a therapeutic standpoint, none of our disinfection approaches currently, be it hypochlorite, calcium hydroxide, any of these, none of these target intracellular pathogens. You can kill some of the biofilm bacteria, yes, but we know very well right now that some bacteria will always survive treatment. So no matter, even if you're going to use an 8% bleach followed by calcium hydroxide, you would still have some remnant bacteria in the root canal space. So this could rapidly enter into stem cells the minute we evoke bleeding inside for regeneration. So unless we design or discover newer antimicrobials that can kill these bacteria that go into stem cells probably and kill the bacteria selectively, which none of the current approaches can do, then that could likely be paradigm changes in how we perform regenerative therapies.
Yeah, that's really fascinating. I mean, there's so many different levels to this discussion. Clearly, I mean, we know from the example, as you mentioned in perio, you have P. gingivalis that has been found in epithelial cells lining the socket, as well as elsewhere in the body in all kinds of different places. The macrophages that do get these other types of cells is true as well. My understanding, though, was that some of these cells that would become persister cells inside these some of these bacteria that become persisters, that's kind of a mechanism to evade antimicrobials in many ways. Because, right, for example, if you give antibiotics to some people, the bacteria could go in a dormant phase into the cell just to evade the bacteria, such as penicillin, that requires multiplication, so it evades that. So how can you, and it's interesting because apparently these bacteria are somehow, as you said, they're evading the immune system by being inside the cell, and they're not putting out any of their markers on the cell surface that could trigger the immune system to come and kill those cells, right? I mean, you could die by apoptosis, or you could actually die by your immune system attacking it, your lymphocytes or other macrophages or whatever, to come in and kill them. So How is that going to be possible to kill them without, with them hiding so well?
Oh, brilliant. So there are two parts of this question, right? One is, what are these bacteria doing? So these bacteria are not passively entering these cells. They are actively using membrane modification mechanisms, which we did report in our paper, to go into these stem cells. And that depends on the type of bacterium. If you take Enterococcus faecalis, a gram-positive bacterium, and Fusobacterium, a typical periodontal gram-negative pathogen, they use very distinct mechanisms to enter into these stem cells. So it's not just a passive random process. So it's a well thought out process by these bacteria to go modify these stem cell membranes and then enter inside. So once they go inside, what could we do? Now, naturally, what we expect is that these cells would commit a suicidal mechanism and would kill the bacteria and themselves, but that's not happening because the bacteria are actively shutting down pathways that would prevent their recognition. What can we do to do this? Now, about 2 years ago, we published a paper in the International Endodontic Journal where we designed a specific antimicrobial peptide delivery system, which we could use to take antimicrobial peptides into macrophages and kill intracellular bacteria. So what we saw was that these peptides don't kill the macrophage. They selectively go into the macrophage and kill only the bacteria. So we have a mechanism in place. So we have a peptide in place. And we also have those membrane permeable peptide conjugates, which we know can go into these cells and then kill them. Right. So that's work in progress. So we are currently testing if those can be used for entering into the stem cells. But beyond that, we have a bigger vision. We have been working. I mean, of course, I cannot speak too much about that because it's still work in progress. We don't have information yet. We're seeing if there are ways by which we can even prevent these bacteria from even going into cells. Could there be supplementary disinfection approaches that we do? You've heard my talks before, so you likely know what we've been doing. Could we use supplementary disinfection approaches that could actually prevent these bacteria from even internalizing pathogens, internalizing host cells? There is a possibility. There are possibilities there. And once we have some conclusive data, and I think that is something which would definitively change how we perform regenerative treatments.
Well, I think, I mean, in that effect, I think this goes far beyond regeneration and endodontics. I mean, that's a universal problem elsewhere in the body with all kinds of bacteria, dermatological, all kinds of different places in which you have bacterial invasion of the cells themselves, and whether it's stem cells or even other cells, as we talked about. So that could be, if there is such mechanism, that could be a big leap forward overall. It's very interesting, though, because it really, I mean, and for those of you who may have not kind of fully read the paper yet, the idea is really these microbes, in endodontics, clinical endodontics is about cleaning the inside of the root canal, shaping it, and we know that there might be still some little bit left in there. That's why we obturate to prevent any of the bacteria coming out. But this particular hypothesis, which is based on the microbes that would be even leaving the root canal space outside to the periapical issues, invading specific cells, whether historically, as we know, in macrophages or in this particular case in the stem cells, can go into a persister state or become a protected reservoir for coming back later on. Because we've all done these beautiful root canals that later on you wonder, well, why is it not healing and why is there still persistence of a problem? And this is an issue. And for those who may be holistically thinking and saying root canals don't work, would be a similar problem with implants. So it would be a persisting of bacteria. It's not inside the tooth or inside the space. It's actually outside on the cells. So periodontics, endodontics, all of these it would apply to, for whether it's root canals or implants. It would be a universal problem. So it has nothing to do with root canals. But this is a very, really interesting conversation because that's how that bacteria goes in there. Almost, the equivalent of that is forming spores, right, in a surface. But you can think of it as forming a spore inside a cell, a human cell. So you have the outside of the cells protecting it from being recognized. And then it comes out. So this is really a fascinating conversation. I think you've then it's a great kind of entree, and that's why I wanted to talk to you, because I feel like this is a whole new approach to understanding failure, as you mentioned yourself. So what do you think the practical implications of these are on a mechanistic side of our cleaning and shaping and disinfection, and in terms of prognostic side, in terms of what we decide for a tooth, and any future things that you think would be around the corner to help address this issue?
All right. So first thing I want to agree with what you said in terms of implant failures, because what we're speaking is not a root canal specific mechanism. If you talk about periimplantitis, this is a fantastic mechanism by which organisms can survive there and cause persistent damage and come out and kill all that's surrounding that, right? In terms of how we could revolutionize our therapeutic approaches, over the past maybe 5-6 years, we've been working with mechanisms by which we can shut down microbial communication. So one of the works that I'm very proud of from our group is developing antimicrobials that prevent bacteria from talking to each other. So we know that bacteria talk to each other by producing chemicals, and that's a mechanism called quorum sensing. So quorum sensing is a whole mechanism by which bacteria come together, form a biofilm. It also regulates many aspects of virulence, about how they cause damage, how they cause tissue breakdown, many different things. So quorum sensing inhibitors are something which we have been developing over the past years, which can prevent microorganisms from repopulating and forming new biofilms. So they can really prevent reinfection, because if these organisms never come together and form a biofilm, they don't have a critical mass to cause damage. So even from a routine reinfection standpoint, quorum sensing inhibitors as a coating prior to root canal obturation could be a good step forward, which is what we are currently working on in terms of developing that. Could these also play a role in terms of preventing microbial invasion into mammalian cells? Possibly. Some of our data do support that, but that is something which is still work in progress. But it's likely that these anti-virulence approaches, if we develop them into a clinical workflow, and if these become dentine coatings prior to root fillings or prior to regenerative therapies, it's possible that when our stem cells come inside, the bacteria are not able to go in and invade into these cells because these pathways are being shut down. So that's something which we think could be a major leap in how we perform these treatments.
Yeah, it's amazing. I mean, it seems like we're entering a new era in which endodontics is moving away from a mechanistic side of endodontic therapy, radiographically based and purely mechanical, to one in which we're trying to appreciate the biological basis of failure and trying to address that. I mean, although your study, I think, was primarily on individual microbial infections in vitro for people to understand that, it would also be interesting to see how a polymicrobial infection in the form of a biofilm would react under similar circumstances. And also, I think the one other issue was that, I mean, to what extent will the immune system address those SCAP cells or the stem cells that have been infected? That, I don't think you had done that yet. Is that something that would be possibly the next step, to use an animal model and to study that?
So there are two parts here. The first thing is in terms of biofilm versus free-floating bacteria. The work that we currently reported is on monospecies biofilms, and we also report that biofilm cells invade far more efficiently than planktonic cells. So we know that already. In a polymicrobial biofilm, would certain organisms maybe they would invade better than monospecies ones? That's something which we have data on, but we've not published yet. So if you take a polymicrobial culture, some organisms would preferentially go into these stem cells than others. And that, I mean, I would not be surprised with that finding. I mean, because that may not change paradigms, because we know that some organisms are better invaders than others. How would the host immune system respond to this is currently a manuscript we are writing up.
Okay.
So that was part of our Foundation for Endodontics funding, and we are almost finishing up the project, and we are writing that up. So we are seeing how, not in an animal model, but in a very well controlled immune system stem cell co-culture model that we have developed, we are seeing how these intracellular bacteria would influence the macrophages are not able to detect the presence of intracellular bacteria. So what we are currently doing is going in to do a bit more of fundamental science assays, which may not be clinically important, but then for us to understand how exactly is that possible? How is that the immune system is not recognizing the presence of an intracellular pathogen?
Right.
Is it doing something, right? So we're looking at that currently. Is this possible in an animal model? It is possible. The problem with animal models is that it's impossible to get a consistent and reproducible intracellular infection in something like a root canal system. People have done this with mouse models on skin infections. That's
Right.
possible. But if you were to take a mouse model of an intracellular infection in the periapical region, that is going to be a challenge in getting standardized infection across multiple different animals. And also the cost factor is a big issue, right?
Right.
When we look at studies, using mouse models, you want to use 40, 50 mice, you would need several millions of dollars for studies like of that nature.
Right.
And I don't think we have funding to that level currently for endodontic studies. And which is why many groups are working on what we called new approach methodologies, which our group has been championing as well, where we develop more clinically representative cell co-culture models or organotypic models, any of this which could reasonably replicate what happens in vivo without using an actual animal. And I'm very pro new approach methodology models.
Yeah. That's terrific. I'm glad to hear that. Another reason why I was so interested in this paper is because just by reading it, it reminded me of something we all know who studied biology, is that this type of persister cell inside a kind of a eukaryotic cell is how eukaryotes started, right? I mean, we knew that we had prokaryotic cells as the first form of life, which was single cell organisms for so long. And then at some point, about a billion or so years ago, you ended up having one microbe and enter and find a symbiotic relationship with a prokaryote, and that created the eukaryotic cells. It's now we know that as the mitochondria, right? The mitochondria, one of the theories is that it was a symbiotic microbe that invaded, and that created the complexity of life we have, which is fascinating that this type of relationship could exist in which it could be beneficial. In this particular case, we were talking about non-beneficial parasitic and other things of it, but it's fascinating that it only happened once according to the mitochondria theory. But yet we're having these on a daily basis happen, and it still hasn't found a beneficial way that it could propagate moving forward. It's really fascinating stuff, and it's terrific. I know you have to go, and I don't want to keep you too long, but keep on doing the stuff you're doing. It's just really terrific, and it's just great work. So
thank you so much. So once we have the other 3 or 4 papers in pipeline published, we should really have a chat again to discuss about those.
I can't wait for that. Well, Dr. Neelakantan, thank you so much for walking us through the science and the thinking behind this work. If this paper teaches us anything, is that regenerative endodontics may be limited not just by biology, but by microbiology at a level that we haven't really fully addressed in the past. So intracellular persistence challenges are assumptions about disinfection, about failure, and about what truly regenerative therapy really requires. I encourage you guys to all read the full paper and also to reflect how these findings may fit into your understanding of regenerative therapy. This has been Inside the Paper here on EndosKool. I'm Ali Nasseh, and I'll see you in the next video.